Welcome to Ajad Bioacademy. Today I will discuss cardiac muscle contraction. I will try to explain the whole procedure of cardiac muscle contraction. Okay, so before going to the details, just I will I can write the heading cardiac muscle contraction. Okay, so the cardiac muscle is somewhat different from other muscle because within our body three types of muscle, that is skeletal muscle, smooth muscle, and cardiac muscle. So today's main focusing matter is cardiac muscle contraction. So how cardiac muscle is different and what is the structure of cardiac muscle, anatomy of cardiac muscle, that I will explain first and roughly, and then I will go for the contraction procedure. So come on here. So this is a simple membrane of the cardiac muscle, cardiac cell, and the name of the cardiac cell membrane is sarcolemma. Okay, so this is the membrane. So inside the cell, multiple cellular organelles will be there. So here within the sarcolemma, that means within the cardiac muscle, cardiac cell, several myofibrils are there. Myofibril. Okay, these are the myofibril. And myofibril actually made up of multiple number of sarcomere. Okay, and each sarcomere contain a nucleus like that. These are the sarcomere. Okay, that means this is one sarcomere, this is one sarcomere, this is one sarcomere. So this is sarcomere, and these are myofibril. Okay, so the sarcomere is one of the major part of the cardiac cell that will make contraction and relaxation. Okay, so what will be within the sarcomere? So if you just make it elaborate, then it will be easy to understand. Okay, so sarcomere is basically made up of a filament, that is thick filament and thin filament, that is actin and myosin. Okay, so how they are organized within the sarcomere, okay, then I will just now try to draw. So this is a Z line or G line, and here you can say this one is also a G line. Okay, here on here the filament, thin filament are there, thin filament. Okay, the name of the filament is actin. Name of the filament is actin. Okay, so the name of the filament is actin, and this is G line. Okay, this is the thin filament, and among them thick filament are there. Thick filament. So this is thick filament. This is thick filament. Okay, you can see this is thick filament. Thick filament. And thick filament name is called myosin. And myosin has some myosin head. Myosin head. Head, the color is different. Like that. Okay, so this is the Z line, and here actin myosin, that portion which contain actin and myosin. So this from that area to that area, you can say A band. Okay, and here you can differentiate another things. There only actin is there. So from here to there. Here another region. Okay, so here, and this is called I band. I band. And within that, the main point here, a special type of protein, that is C protein, M protein, and different types of protein made up of a same center point, middle point, and this is called M line, or you can say M zone. Okay, and if you just look carefully, then you will get another zone, that is another line is that area, only present in myosin, and that region is called H band. Okay, or H zone. So that way actin and myosin is just assembled and form a sarcomere. And you know, this is the sarcolemma. So within that part, different things, that is, you can say this is endoplasmic reticulum. You can say this is your mitochondria, like that. So different organelles are there. So what will happen if cardiac muscle contraction is necessary? Then what will happen? The actin myosin will be just try to come towards the M zone. Okay, then that will cause contraction. That means actin and myosin should be combined to each other and make a complex. And how it will occur? That is the question. So for the contraction, what is necessary? Necessary things is action potential and their rearrangement. Okay, so action potential means electricity. So what will happen? You know, action potential is generated within your SA node and then AV node, and then it will just circulate bundle of phase and Purkinje fiber. That means all the junctional tissues, and that will make cardiac contraction and relaxation. So for the cardiac contraction, action potential is very much important. So if you just think here, this is the G zone, that part, and I'm just after, just beside the G zone, here from the sarcolemma, different gap junction there, and you can think they are from the part of the membrane, another tube-like protrude inside the cardiac cell, that is called T-tubule. So that will be present, you can think here, like that. Okay, and you can see that is the membrane. So this is the sarcolemma, and this is the T-tubule. Okay, and action potential will reach. So firstly, how action potential will produce? So action potential, you know, the entry of sodium, it will cause depolarization, and this is the first active process of action potential. So when the entry of sodium, action potential will reach, and within the T-tube, here on a special type of channel is there. So this channel name is called, you can say this is called L-type calcium channel, and you can also call it DHPR. This special calcium channel is named L-type calcium channel, or you can say dihydropyridine receptor, that is DHPR. Okay, so when action potential will reach there, by the influence of action potential, that calcium channel will open. And you know, in T-tubule carry some extent of calcium. Okay, so calcium will enter. Where it will enter? Now I will say, already I said here, like here, sarcoplasmic reticulum, especially for cardiac muscle, sarcoplasmic reticulum are there. So I will just draw this line in different way. Okay, so this is your T-tubule, and here is just incorporated into the cell. So over there, one sarcoplasmic reticulum is present. So sarcoplasmic reticulum basically carry calcium. Okay, so you can say the sarcoplasmic reticulum is the storekeeper of calcium, and these are the calcium. Okay, so what will happen when action potential reach to the terminal of the T-tubule, and there a special type of channel is present, and that is called, already I said dihydropyridine receptor, that is called L-type calcium channel. Okay, so already I said here it will contain a certain extent of calcium, so that would be like that, and calcium will enter from there into the cell, that is in cytoplasm. Okay, then what will happen? The sarcoplasmic reticulum carry a special type of a receptor. A special type of receptor, and you can say this receptor name is called ryanodine receptor. That is ROR, ryanodine receptor. That ryanodine receptor is basically calcium dependent calcium releasing receptor, because when after influence of action potential, when L-type calcium channel open and calcium will enter, that calcium will bind with the receptor and it will make it open, and then through this receptor calcium will come out. Okay, calcium will come out. This is calcium, and that calcium actually black color already I said inside, so that should be blood color. So that is calcium, and that calcium, that means intracellular calcium level will increase. Okay, what will happen next? So intracellular calcium level will increase, and that calcium will contribute for the contraction. What I will just make in different category, like this actin myosin matter. So here actin is open almost. I just tried to show, but now I will show it in different way. Like actin is basically just composed of like that. Okay. Okay, that means like that. The double strand helix-like structure, and it's composed of monomer protein. Okay, so this is actin, and like that, that's actin. But this in normal state, actin will not be free like that. What will happen? In every 7 monomer, a special type of troponin protein there. Troponin protein. Okay, troponin protein. So here you can say troponin protein, and here troponin protein. So in that way, multiple number of troponin protein complex are there in every 7 monomer interval, and you can roughly say 5.6 nanometer distance. Okay, and that is not enough. Another things, that protein will just make the actin is just blocked by a special type of chain protein. And what would be like that? That is like that. So this is okay. Another one is that. Okay. That means if you think this is your actin protein chain, and this is your binding site, and it is blocked by a special chain protein that is called Tropomyosin. Okay, and this is called actin. Okay, so this is tropomyosin and this is actin. We can write in blue color. So this is actin. Okay. Now what will happen? Actin is completely blocked by the tropomyosin with the help of troponin complex. It must bind with the myosin to make contraction. Okay, so like that. So now I will draw. So I can write here the tropomyosin. So here is a thick filament that is myosin. Okay, and myosin has myosin head. Myosin head. Okay, and normally what will happen? This troponin complex has three types of troponin. Already I tried to show you in three different color. That is, I can say this black one is troponin C. You can say the red one is the troponin I, and blue one is troponin T. Okay, and it has several roles. So what will happen? Our main target is to make bind with actin with myosin, and it will make a actin myosin complex, then contraction. Okay, so how come? Now that calcium will come off here to bind with the troponin C. If you want to just remember very easily, so C you can consider calcium. So calcium binding troponin C. So calcium will bind with the troponin C, and it will change the conformation of the complex. And what will happen? It will just remove the inhibitory function of the troponin I, and it will make the changes of troponin T, and that troponin T will maintain to just remove the tropomyosin or rotate the tropomyosin from the active binding site of the actin. Okay, just like that, already I said. So in that way, so what will happen? When calcium will bind with the troponin C, then conformational change will occur, and troponin I, the function of troponin I is to inhibit the binding of actin and myosin by keeping the position of tropomyosin on the surface of actin, and it will be maintained by the troponin T. Okay, so when calcium will bind with the troponin C, then conformational change occur, and that tropomyosin will just rotate, and it will open the like that, and it will open the active binding site of the actin. That means it will just rotate and change its position. That means the actin will be free. Actin will be free like that. That is actin binding site, free active binding site. At that time, what will happen? ATP will be hydrolyzed by the ATPase activity. That is, ATP will be hydrolyzed, and it will form ADP. ADP. The color problem, I will just try to make it clear. That is, ATP will make ADP and one inorganic phosphate release, and that ADP will bind with the myosin head. Okay, and during the hydrolysis of ATP, energy will be produced, and that energy will be used when the ATP bonded myosin head will be ready to bind with the free actin. Okay, by the binding of calcium with the troponin C, tropomyosin will be detached and actin will be free. And with the actin, free actin binding site, myosin head will bind by the help of ADP and energy that will come from the hydrolysis of ATP. Okay, so what will happen when energy will produce an ADP containing head will bind with the actin? How? It will change its conformation and it will stretch like that, and it will just search the binding site of actin. Okay, if I just draw here, so this is your major color problem again and again I am making. So this is actin, like free actin. Okay, and the myosin head was like in that position in normal state, and when ATP will bind and ADP containing, it will change the conformation, like it will twist and it will bind with that. Okay, from that position it will be in that position with containing ADP and IP from the ATP. When ATP will be hydrolyzed and it will make ADP, and that will be attached with the head of the myosin, and that myosin will change the position to bind with the actin, like stretching like that. And then what will happen? It will just snatch it towards the center of the middle of the actin myosin complex. Okay, already sarcomere. In the sarcomere, the middle part, M line towards in that part or in that part, that will try to stretch. Okay, so stretching will be occurred by that way. Okay, and this stretching is called power stroke. Okay, this is the power stroke. In that way, actin myosin will make a actin myosin complex, and that will cause contraction. Okay, so contraction is just happening with the help of calcium, intracellular calcium, bind with the troponin C, and troponin I will be inhibited and troponin T will just make the maintenance of that part. Okay, another thing is that, and that will completely happening by the help of action potential. But your heart is just working with your body demand. Suppose you are afraid, you are in a hurry, or you are taking exercise, so your heart should work more and more. That means heart rate will be increased. So your noradrenaline, that means sympathetic neurotransmitter, will help you. So in that way, from the membrane signaling pathway, PKA, that is protein kinase A, will produce, by beta 1 receptor signaling pathway, and that PKA also can contribute the contraction. That PKA can phosphorylate the troponin I. and it will remove the inhibitory function of the troponin I. Then what will happen? It will facilitate more calcium binding to the troponin C. Okay, and then finally that will also occur. And also that PK has another function, is PK can just phosphorylate the ryanodine receptor to release more calcium inside the cell, that means intercellular calcium increasing. Okay, those are the contractile contribution. But suppose if you think more contraction occur, so what will happen? If excessively contraction occur, then cardiac problem may arise, or your arrhythmia problem may arise. So in that case, PK can play a role. That means PK has dual role. At that time, the sarcoplasmic reticulum membrane has another receptor, that is SCR CA. Okay, SCRCA. So that protein is responsible for entry of reuptake of calcium. Okay, and here one inhibitory protein is there. Inhibitory protein are there, and that inhibitory protein name is phospholamban here. That will inhibit the ACR protein transporter, that is channel, the phospholamban, and PK will just phosphorylate the phospholamban. The inhibitory function will decrease, and that channel will work properly to reuptake excessive calcium through this way, and that will prevent the excessive contraction. Okay, this is the different things. So you can say this is somewhat unnecessary here. So overall, you can say now the overall, this is the cardiac muscle structure, and this is the sarcomere internal structure, that is thin and thick myofibril. Thick and thin the filaments, actin and myosin. This is the Z line, already I mentioned. So here is the whole process. Okay, so thank you very much.